This is TWIS, This Week in Science, episode number 560, recorded on Wednesday, March 30th, 2016. Science, no foolin'. Hey everyone, I am Dr. Kiki, and tonight on This Week in Science, we are going to fill your head with a bloodthirsty prairie dog, poop for puppies, and fridge lasers. But first...
Disclaimer, disclaimer, disclaimer. The first time human ancestors left the tree to go walking about in the wide open world, they must have been afraid of everything, or everything must have been afraid of them, or nothing much noticed them until it was much too late, and humanity had spread worldwide. In any case, the few things that were noticed or not, caused or overcame fear, had one thing in common. Knowledge. Our ancestors' ability to acquire knowledge made them powerful enough to set out, even with 8 brains, and conquer a planet. And so, in the spirit of our 8-brained heritage, we offer you knowledge so that you may set out from the trees of humanity and conquer the world, or do something great with your day. Yes, it is time once again for This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And good science to you, Justin, Blair, and everyone out there. Welcome, 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 welcome to another episode of This Week in Science. You know what? It's not April Fool's Day today.
No, it is not.
So we're not doing an April Fool's Day show with a lot of fake science. We're just doing a show with a lot of science.
Good, because there was a lot of good science this week.
I know, there's tons. I'm super excited. Lots of great stuff out there. What did I bring? I brought stories about steps to synthetic life, fecal transplants, and Saturn. Justin, what'd you bring?
I've got a handful of human ancestor stories, as the disclaimer might have anticipated you towards.
I believe that with you, with your hands full. You're like, human ancestors, gotcha. I'm going to bring that. Okay. Blair, what'd you bring for the Animal Corner?
I brought the aforementioned bloodthirsty prairie dogs. I also brought some vengeful birds and a couple new ways to save endangered species.
Oh, we like that. I like good ideas on how to save things. It's always nice. Needed and nice. Let's get this show going.
What?
So right off the top, crazy, awesome, what? Blow your head kind of news. Well, Craig Venter is back at it again. So he's been kind of silent, or at least the papers on synthetic biology, synthetic bacteria, creating a synthetic organism, have not been flooding academic journals as they were a couple of years back. It's actually been a couple of years since we've seen a paper from the Venter Institute on synthetic biology, on creating these synthetic organisms. The last paper, they booted up an empty bacterial shell, basically, with a bacterial genome that was an existing bacterial genome, but they printed it and then stuck it into another bacteria's nucleus and said, haha, let's see if it will divide and conquer, and it did. So they showed that they could print these things. If they had a bacterial DNA sequence, they could print the DNA and they could make a bacterium go. But that is not everything they need to know to create a completely de novo bacterial species. what they're trying to figure out and what they've been trying to figure out since then is how to create the most minimal genome possible. And this is something that's really important in the way, in the pathway of creating life from scratch, is what genes are necessary for life to go. What do you need? What kind of instructions do you need for it to start translating itself and making itself reproduce and survive? How does that happen? So they took the bacterium in this new science paper, bacterium with the smallest genome that is known, and this genome is called Mycoplasma mycoides. It's a tiny little bacterium, and they've been working at it, and it hasn't worked. They tried a few different methods, and they didn't work. And so this is why it's been so long that we haven't heard from them for a while. Things have not been working, except they finally said, okay, we're just going to start with our bacterium the way we know it, and then we're just going to start deleting things and see what happens. And so they've been doing a step-by-step iterative process, figuring out which genes of this mycoides genome were necessary. This genome started out with 500 some odd genes, 525 genes actually, and they came up with a genome finally after round after round after round. They call it JCVI, J. Craig Venter Institute, SYN 2.0. So synthetic 2.0. That kind of makes sense for the naming. And so starting from 525 genes, they reduced it down to 478 protein coding genes, and then another 38 genes that were not protein coding but are used in RNA and creating, say, messenger RNA, interference RNA, various kinds of RNA that float around the cell. So another 38 were used for that. And they said, okay, that's great. That's a little smaller. We still don't know what all these genes are for, so let's see if we can get it even smaller, see if we can get rid of some more stuff. So more mutating and making things not work, and they created JCVI SYN 3.0. They got it down to 473 genes. And according to their paper in Science, the 473 genes have genes involved in transcription and translation of proteins. So some of the proteins are necessary for creating the ribosome and getting the ribosome to work. Others are for specifically taking DNA, turning it into RNA, and then getting it over to the ribosome itself. So this whole process, transcription, translation, those proteins are in there. But then there are 149 genes of completely unknown function. 149 genes out of 473, they still don't know what they do at all. But so far, with all the mutations that they have done, 473 genes is the smallest they think that they've been able to get it so far. Of course, they're still mutating it. Of course, they're trying to get it to be smaller if they can. But they're also, the next step is to figure out what these other genes are involved in. So what undiscovered purposes or undiscovered functions are necessary for life. So yeah. So there's some really interesting stuff there. From the conclusion of their abstract, they say that the minimal cell concept appears simple at first glance, but becomes more complex upon close inspection. Yes, it does. In addition to essential and non-essential genes, there are many quasi-essential genes, which are not absolutely critical for viability, but are nevertheless required for robust growth. So the bacteria can live without some of these genes, but they don't grow very well. They don't flourish. So they say, and also they found out, of course, a lot of genes are interlinked, and so the functioning of one gene affects the functioning of another, and so there are these downstream effects. Consequently, during the process of genome minimization, there is a trade-off between genome size and growth rate. JCVI SYN 3.0 is a working approximation of a minimal cellular genome, a compromise between small genome size and a workable growth rate for an experimental organism. It retains almost all the genes that are involved in the synthesis and processing of macromolecules, and then those 149 genes with unknown biological functions. And finally, JCVI 3.0 is a versatile platform for investigating the core functions of life and for exploring whole genome design. So they're back. They're mutating this minimal genome. They are looking to see what genes are essential for life and, like they said, the trade-off between life and growth.
And so they figured out that you need this almost 500 genes to survive. That's the minimal genome. But the fact they don't know what a third of them does, but they know they're essential.
Right.
There's something about that that's fantastic to me, is that we figured out, okay, we really need this.
We don't know what they do. We've taken
No
them
idea.
out, and then the thing dies, but we don't know why it dies.
So,
Something breaks. We don't know what.
And this might not be the only configuration, right? But this is the right. I mean, this is the smallest stable version that they've gotten, at least.
Yes, this is the most stable. Exactly.
Yeah.
Like they said, the best trade-off between it living and not just being dead, and actually growing and not just kind of sitting there going, well, I'm here, but I can't do anything. Yeah, and John Timmer over at Ars Technica, he in his last paragraph of his write-up of this work, he said something that was one of the first thoughts that I had as well, and it's the idea would be to take these cells, and right now the cells that they're using are bathed in this super luxurious nutritional bath, so they don't have to worry about making any of their own nutrients. They don't have to worry about like manufacturing vitamin K or any of the things that they might need to survive. So this is what helps it be a minimal genome. So what if we just started taking stuff out of that bath, taking nutrients away, and seeing what came of it? And then you have actually a system or a platform for some really interesting research into evolution to find out, as John Timmer says, to tell us a lot about how much of what we think of as being typical life is a clear optimization and how much is simply the result of lucky accidents. So it could actually be experimentally checked now, which is pretty cool. Pretty cool. So yes, I'm sure there will be much more on this synthetic biology front from the Venter Institute in the future. They've sped up their process, so maybe it won't be 2 years until we see their next paper come out. Moving from a little small space to outer space, Saturn. We all love Saturn and its beautiful rings, and the Cassini mission has brought us so close to those rings that who would have ever thought that we'd be as close to Saturn as we've been for the past decade? Anyway, researchers have been trying to figure out the dynamics of the rings and the moons around Saturn. How do they form? What's going on there in the gravitational forces that allow them to have formed where they are and to have the orbits that they hold? And there's some really interesting thoughts that start to arise when you just start to think about the gravitational forces of Saturn itself, and then between the different moons. And in our own solar system, we have some really interesting oscillations that occur as a result of gravitational interactions, say, between large planets like Neptune and smaller outer dwarf planets like Pluto. So Pluto is hugged around by Neptune's immense gravitational field. Some of us, like Earth, we have a very stable orbit, but that's only happened as a result of many interactions that occurred millions, hundreds of millions, billions of years ago, actually, when the solar system was formed. So, how much do we know about the stability of the orbits of these moons around Saturn and where they came from? Well, there is a researcher named, and I'm going to probably pronounce his name incorrectly, Matija Chuk, and is a lead author of this research paper published in Astrophysical Journal. And it's authored by a bunch of scientists from the SETI Institute and Southwest Research Institute. And they're using observations from the Cassini spacecraft of the interior moons of Saturn and the rings. And what they have concluded from their analysis is that the inner moons are super young. They're nowhere near the age of the planet. Like, our moon is thought to have formed very shortly after the formation of our planet. That it just had, our planet was formed, and then boom, big object came and smacked us one, and then the moon flew off and then stayed because we had such a nice gravitational interaction. Luck would have it. They think, however, that the inner moons of Saturn might have gotten their start only about 100 million years ago.
Wow. So there's dinosaurs.
The dinosaurs. Yes, the dinosaurs looking at the sky, seeing brilliant Saturn up in the night sky. We're watching the moons form, like Enceladus forming 100 million years ago.
So any ideas what brought this about? Because there's plenty of moons there. Is it that other moons that were already there bumped into each other and then had to reform new moons, or is it an asteroid hit kind of a thing? How did they get these new moons? Or has Saturn just been slower than some planets in sort of forming its moons? That these were other dust rings or something that were floating around out there.
Yeah, so the idea is that maybe they had been little moons and then they got broken up into rings, and that those rings again formed into moons. So there's kind of been a back and forth over several billion years, but the most recent is that there were there were rings of debris around Saturn that formed up into these moons around 100 million years ago as a result of the orbital dynamics and particular oscillations that occurred at certain resonant frequencies around the planet of Saturn.
So then the rings that Saturn still have might just be like a sweet spot, like this weird sweet spot that should have formed a moon. Anywhere else would have formed a moon, but in Saturn they're either just sticking around, or maybe this is how a moon starts. It starts as a ring, and we've just seen all the planets that have moons should have rings, or did once, and now we're just sort of seeing one that didn't form.
Yeah, so that there would probably some collisions that occurred, that there was instability at some point. There was they kind of started shaking, if you've ever watched like waves start going and in pattern and they kind of get out of sync and the instability grows, and that probably ran some little moons into each other, crash, debris all over the place, and then soon after that the newer moons of the inner Saturnian system got started. So it's a really interesting idea, and one of the things that comes from it, though, is that depending on the stability of the system now, and I don't know if the system is now stable, it may yet deteriorate again. It happens to be stable as we're looking at it currently. Because there are tidal forces and the inner moons are moving away from Saturn very quickly. So since they're moving away from Saturn so quickly, maybe there could still be some disturbance in their system that could lead to more crashes and bangs and debris fields and more new moons later. Additionally, though, it suggests that Enceladus is very young. And Enceladus, being this wonderful water-carrying moon, we've seen from these beautiful water jets that shoot out from the stripes on Enceladus, that there's water, ocean of water beneath the frozen surface of the moon of Enceladus. And if there is water, we've thought, hey, maybe there could be life, right? It's like, oh, with water comes the possibility. But if it's only 100 million years old, within the time of dinosaurs, is that long enough for life to have gotten a start? So now people are wondering about this idea that Enceladus might hold life in it.
Yeah, it seems a little young, right? Now it sort of throws off that whole time scale upon which you think these things could, by chance or happenstance or conditions, even get going.
Exactly. Yeah. So we don't know. I mean, if life was, populated our solar system on comets, that happened a long time ago. If the building blocks were brought and life got its start just from the right conditions happening, that happened still a long time ago.
We think. Again, but we don't really know the conditions exactly. But, yeah. This doesn't look like it. This doesn't have anything going for it that would then say, like, well, maybe.
So Enceladus, in addition to having these wonderful cracks in its surface from the tidal forces, the gravitational forces of its orbital path around Saturn, it has these water jets that are brilliant in relief against space. And researchers have been trying to figure out how exactly these wonderful water jets work. So some researchers have published a study this week that's a model of how they work. So wonderful computer model to make the whole thing work. It's published in the Proceedings of the National Academy of Sciences. And the take-home message from it is that they've been able to recreate one aspect of the geyser's behavior, which is that there's a 5-hour delay between the tidal forces pulling the cracks in the ice apart to their maximum width and the peak of how much water sprays out, how much erupted material comes out. It's about a 5-hour delay. And so their model was able to do that. It's pretty awesome. And the model has discovered that this occurs because there's turbulent flow in the crack between the surface of the ice and the ocean underneath it. And the turbulent flow down that crack, it's like spiraling and turbulent. If you can imagine lots of, like a rapids with lots of turbulence in it. The turbulent flow slows down the refilling of these slots with water as the tidal forces change. And so the tidal forces are changing, the crack width is changing during that time, but the amount of water that is held in place in those cracks and is getting pushed out is not changing as rapidly. So the other thing is that the model was unable to capture, there's this a 5-fold difference in the amount of material that erupts at different points in the orbital cycle. So as this moon is orbiting around Saturn, there are times when it's just spewing tons of water into the air, and then other times when it's really not. 5-time difference. So they were not able to narrow that down with the model, so they're still working on it. But basically it's, we've got these cracks and they're being pulled open, and then the tidal forces are shoving them shut again, and it's kind of like a pipe, if you could, or a hose. If you take the hose and squeeze it shut and then open it up again, it changes the amount of water that comes out. And so they're trying to just figure out the mechanics of how it works. But the big thing about this is it does tell us that these jets, even though Enceladus may only be 100 million years old, these jets, due to the tidal forces that are at play, could keep the pressure up, to keep it going for up to 1 million years. So the pressure and the heat that's generated by the tidal forces tweaking the surface of this little moon can create all that energy and keep a sustained water flow for 1 million years. So we had plenty of time to send Cassini out and to image the beautiful pictures that we have. Right? Yeah. Yeah. So, this is This Week in Science. Moving on up. Justin, tell us some human history.
Some human history. Well, this is actually an update on this one. This is Homo Floresiensis, the so-called Hobbit folk of Indonesia, have been redated. International team of scientists investigated the original site and have published their findings. This is the site, the little Hobbit folk in Indonesia found on the island. They're only a couple feet tall, have these tiny, tiny chimp-sized brains, and yet have tools, and were thought to coexist because the modern humans got there 50,000-ish years ago. This is saying that the site is no longer thought to now be between 13 and 11,000 years old, but is much older. The most recent Hobbit skeletal remains, they say, occurred around 60,000 years. Evidence of their simple stone tools continue to about 50,000 years ago, and after this, there's nothing to indicate the little Hobbit people persisted, which would put their disappearance pretty much in accordance with the earliest modern human settlers' arrival on the islands, and removes the tens of thousands of years of cohabitation, or at least co-settlement of the island. The site, they say, is large and complex. The original excavators dug only a tiny portion of it. Years of further excavation have led to much clearer understanding of the order of the archaeological layers, and it is now evident that when the original team collected samples for dating, they did so in the main layer containing the Hobbit bones, but it was mistakenly from an overlying layer that is similar in composition but far, far younger. So, Homo Floresiensis, much older than we had initially thought they were.
Which is really interesting. I read something along with this. I mean, if we go 50 to 60,000 years ago, around the same time, humans were making our way down to Australia. That's when a lot of animals were going extinct. So could there potentially be a link between the spread of Homo sapien and the extinction of Homo floresiensis?
Very likely.
Homo sapien killing off a whole bunch of stuff as we moved and overhunted, and
yeah,
maybe the Hobbit as well.
And right, and from the Hobbit's perspective, being that they topped out at about 3 feet tall.
They were little people.
Yeah, it would be as though suddenly the planet Earth were invaded by a much advanced race of humans that were 10 feet, 11 feet tall, and much smarter than us.
Well, size isn't everything.
Well, the brains are bigger too. We were much smarter. We had better tools, craftier.
No, they had tools. They were pretty advanced, right? A nice advanced little civilization.
Sure, I guess they had some primitive tools,
and they had just enough brain for their 3-foot size, and probably much more than their 3-foot size even really needed.
And a lot of these animals that humanity encountered hadn't weren't from the African plains and jungles previously, hadn't been brought up in a place where you've really got to fight and hunt, perhaps.
Maybe,
I could sort of, I'm surprised there's pandas, right? The first humans that came across pandas probably could have just taken them right out.
People just didn't want
We
to
were
go through the
just too
bamboo
darn
forest.
cute.
They were too cute. We did, for all of the failings of early humans, we did appreciate cuteness.
Yeah.
But yeah, anytime we got to an island where there were flightless birds, they pretty much disappeared in our wake.
Unless they were
Mhm.
terrifying.
Who were terrifying?
The
birds?
flightless birds, yes.
Oh.
Like the cassowary, we didn't dare try to take down the cassowary. The dodo, all right.
Okay, we'll take down the dodo. You're
And the
not
dodo,
so
apparently,
scary.
we just rounded them up. We would just
Yeah.
sort of herd them like they weren't scared or really impressed with us, and we just herd
Because
them
they've
onto the
never
boat.
But
had
as
to be.
reported on this show, they were not as dumb as we thought.
No, not as dumb as we thought. Just very, already domesticated.
Yeah. Just not afraid of humans, which why would they be? They'd never
No.
seen one before.
Yeah, but they had those big schnozzes. The dodos should have run away the first whiff. First whiff. What does that smell? Ugh.
Yeah. The other story I had in this tip, Australopithecus afarensis, our ape-like, though upright ancestor of Lucy fame, is in the news again. They were alive between 3 to 3.7 million years ago. Fossils are found only in the East African Rift Valley. That is until now, as they have popped up some great distance from it. New fossils from Kenya suggest that Australopithecus afarensis lived far eastward, beyond the Rift Valley. International team of paleontologists led by Emma Mumbua of Mount Kenya University of and Masato Nakasukasa of Kyoto University. I'm overpronunciating things now. Report findings of fossilized teeth and forearm bone from an adult male and two different Australopithecuses from an exposure eroded by the Kantas River in Ongata Rongai, a settlement on the outskirts of Nairobi. So far, all the other Australopithecus afarensis found have been identified in the center of the Rift Valley, explains Nakata Sukasa. Previous discovery in Chad confirmed that our hominid ancestors' distribution covered good parts of Central Africa, but this is the first time an Australopithecus fossil has been found east of the Rift Valley. This has important implications for what we understand about our ancestors' distribution range, namely that Australopithecus could have covered a much greater area. So part of what I think is really the great interesting part of this story lies in the terrain east of the Rift Valley. Stable isotope analysis reveals the Kantas region was humid, but had a plain-like environment with fewer trees compared to the other sites in the Great Rift Valley where afarensis fossils had previously appeared. The hominid must have discovered a suitable habitat in the Kenyan highlands. It seems that afarensis was good at adapting to varying environments, says Nakata Sukasa. So adapting, yes, and important because Australopithecus afarensis is such a hybrid of ape and man. Upright, yes, but also absolutely they were built for arboreal environment. So to find them in the they were definitely climbers. They still had all the apish curvatures and wrist bone strengths and developments for spending a good part of their time in the trees. So not only did they adapt to this new environment, but this is to find them in a less wooded, more plains-like setting is really setting the stage for the evolution to come. Right? This is when the ape man left the cover of the trees and goes out into the plains and starts to live much more of a modern human-esque way. So very interesting. The team also turned up masses of mammal fossils, including a few that probably belong to a new species but previously undiscovered of bovids or baboons. The site was first noted in a 1991 geological survey. At that time, a farmer had told those doing the surveying that their family had come across a number of fossilized bones back in the 70s. But they didn't recognize the importance. But following the airing of Kenyan television programs on paleontological research, locals started to appreciate these fossils, and they started to identify sites around the area where these fossils could be found. So it was really the local population there that after watching some of this, probably a Nova special of some sort, BBC special, right, said, oh yeah, we've got those. They're all over the place. Come take a look.
Used to make xylophones out of them. Now let's do something else with them.
I just think that's an amazing point to show how important science communications are. The how people can be a part of discovering our own human history if they only know what to look for or what is important in a particular story that's being told. And that's just awesome. Fantastic. Yeah. We need more science for television. Is it time for more science from Blair's Animal Corner?
She loves smart creatures, cry that stuff. Biped, millipede, no pet at all. Wanna hear about animals? She's your girl. Except for giant pandas and squirrels.
What you got, Blair?
Oh, I have some chilling news about a darling of the biological community. Of course, I'm talking about prairie dogs. They're adorable. They're little rodents. They live together. They make colonies. They call out to each other if there's danger. They're harmless little herbivores. Well, it turns out they're not so harmless.
Well, didn't you report a little while ago about prairie dogs? Was it prairie dogs killing other endangered species or something?
Oh my goodness, I don't.
Or was it another animal that was killing prairie dogs? What was the story?
Might have been that. Yeah.
It was It was another animal that was killing prairie dogs.
Killing prairie dogs. That's right.
That's what it was. Okay.
So prairie dogs, they have had population troubles because they're a prey animal. But a recent pair of researchers from the University of Maryland and the University of Tulsa found white-tailed prairie dogs living on the North American prairie. They found them doing some interesting things. So they had a long time study. It was covering the years 2003 to 2012. And they were sitting in trees watching the activities of prairie dogs. 4 years into the study, so this is in 2007, they witnessed in horror a prairie dog killing a ground squirrel. This was the first time ever that there has been an observation of a mammalian herbivore killing another herbivore without eating it. Yeah. So this was weird. Mammalian herbivore killing another mammalian herbivore. There was no necessary consumption of nutrients. Why on earth would they be doing this? Well, over the course of the rest of their study, from 2007 to 2012, they continued to watch out for this behavior, now knowing that it was possible. And they witnessed 101 instances of prairie dogs intentionally killing ground squirrels. And they noticed another 62 squirrel carcasses that looked like they were also the victims of prairie dog attacks. What's more, they identified 47 individual prairie dogs killing the squirrels. So 162 potential squirrel murders and only 47 individuals, 11 males and 36 females. And 19 of the 47 killed multiple times. And I did some math on that, which on these 101 confirmed killings, there were 47 individuals that did the killing, but only 19 of them were repeats. That means that those 19 animals killed 3 to 4 times each in just a few years. So this is a recurring phenomenon. And researchers watching them would watch the interaction beforehand, see what was going on with the resources. They weren't killing them because they were a threat. Prairie dogs were killing squirrels because they were competitors for resources. They eat the same food, and in many cases, the squirrels actually take over abandoned prairie dog tunnels. So the prairie dogs are saying, nah. What's more, they found that mother prairie dogs who killed squirrels tended to have more offspring than did ones that did not kill squirrels, and that their overall quote, fitness levels, something that they were addressing in this research, were much higher than non-killing females.
Wow.
Yeah.
So murderers did better.
Right. So they killed squirrels. You could kind of jump to the conclusion that by killing the squirrels, they're reducing competition in their territory. Therefore, resources come easier. Therefore, they raise more babies. Right? Now, what I think is particularly so interesting about this study is that we've been studying prairie dogs for a really long time. So if we haven't noticed this until now, that means there is a high potential that there are other herbivores doing this that we haven't figured out yet. Just last year, I think, I reported on deer eating meat habitually in the wild during winter when they can't find a lot of plants. They eat meat.
Right.
So,
but
what
that's
we
for
call,
a nutrient purpose,
right?
As
so
opposed
that's
to just for resource competition. I mean, it's a big leap to think that there's a cognitive process going on in there that's saying, because they're not killing their fellow prairie dogs who are also competing for resources.
Right.
So there's something really interesting going on in the prairie dog brain to lead them to this action. There's something really interesting going on.
Eliminate the competition at all costs.
But only the non-con-specific competition.
Correct. Yeah.
Fascinating.
Well, so these are also animals that live in colonies. So then you have to wonder if they came across prairie dogs from colonies that weren't theirs.
Right, other colonies. That's a good question.
Would they then attack? So this has opened an entire new field of research about prairie dogs, but definitely also for other mammalian herbivores. This is something that we have to kind of put a closer lens on for all of these species that we might be trying to protect. There's a lot more going on. These little mammalian herbivores are not these cute little helpless creatures. There's other stuff going on. Rodents.
Yeah, and then the question comes, if you're trying to, protect a ground squirrel colony or population because, say, they're locally endangered, what do you do to the prairie dogs that are killing them? Do you start culling the prairie dogs because they're encroaching on the grounds?
Right.
Whoa.
Look, we can't be the world's zookeeper, okay?
Can't be the world's referee.
Right.
Yeah.
I guess we mentioned that this is the first time we've seen it in herbivores. We see this in meat eaters, right? And yeah, at the end of the fight between a lion and a hyena, one of the others is going to get eaten. But that's because after the fight ended, the other one's going, oh, look, all of a sudden there's food here. Like, it's, it just sort of happens that they also wouldn't be opposed to eating them, but they weren't hunting them for food. They were they were fighting for the same context, fighting over hunting grounds, fighting over the access to the resources.
Right. The question here, though, is that with a lion and a hyena, let's say your example that you were just talking about, they're expending so much energy in the fight. They get some of it back afterwards by eating what they've killed. With the prairie dogs, they're expending all this energy to kill squirrels and not even attempting to get some of it back.
Right. And to be fair, lions will do this. Male lions will attack, at least from what I've seen, will attack a hyena and kill it and not eat it. So there is some of that. It just also happens if they are hungry. It happens to be.
Oh, this is great.
Whereas if you're not a meat eater, you just you'd have to go foraging again or whatever they do.
But interesting stuff. We have to just more things to be looking out for.
Yes, categorizing things often does not work out. It's kind of back to that age-old story. Speaking of which, another animal that we might not have thought up to its devious tricks, Antarctic birds. So there are these Antarctic birds that they're called
Skuas.
skuas. Yeah, so skuas.
Skuas.
Yeah. So they are a relative of the gull and the auk. If you guys know what auks are, A-U-K.
And if you've ever seen the penguin Happy Feet, the penguin animated movie, the skua is the mean bird that comes after the little Happy Feet character.
Right. That makes perfect sense. I didn't even think about that. Yeah. So the skuas, they are Antarctic species that until there were research stations in Antarctica, these species never saw humans. However, now researchers have figured out that these birds can identify specific humans and hold a grudge. So these researchers have to check the bird nests once a week to monitor breeding status, populations, all those kinds of things. And the skuas attacked at closer distances with repeated visits from the researchers. And so once the researchers noticed this, that even if Yang Jiaokan, a PhD student at Inha University, noticed, they said, I had to defend myself against the skuas attack. When I was with other researchers, the birds flew over me and tried to hit me. Even when I changed my field clothes, they followed me. The birds seem to know me no matter what I wear. So after that, they decided to actually do some experiments on this. So the researchers checked the nest once a week, but when they did that, they would have two researchers walk towards the nest, and as soon as the birds took notice of the pair of humans, they would turn and walk in opposite directions. And one of them would be a, quote, nest intruder, so that was someone who has previously assessed nests, and a neutral human, someone who had never assessed the nests before. And when they walked in opposite directions, all 7 pairs of birds followed and tried to attack the previous intruder, never following the neutral human. So they could identify these humans despite what they were wearing. What's really interesting about this is that, as I mentioned before, these birds have never seen humans before the research stations showed up there. So in a very short period of time, birds have figured out how to recognize specific humans. So the question is, was this predisposed, or is this something that was a learned discriminatory behavior over a very, very short amount of time? And I think that it also, it kind of begs the question of, we look at certain animals and we think that they all look very similar. And I've always kind of assumed that looking at a different species, one animal looking at another species, it's kind of hard to tell the individuals of the different species apart until you learn kind of discerning bits. And so a bird being able to identify specific humans that does not have the magnificent cognitive abilities of, say, a jay or a crow, but still, they were able to differentiate humans in such a short amount of time. I think it's really fascinating.
Yeah. I think it may also help that there's not a lot of humans there to differentiate between. A pigeon in New York is gonna see like too many faces and too many humans going by to like remember one of them the next day. Whereas I guess the Arctic bird here in this scenario, right, there's not even another species to stare at and sort of identify. That's
not true. There's species in the Antarctic.
Well,
there's absolutely.
There's lots going on. Well, I will say I understand your point that in New York City, humans are white noise. However, in a place like a very well-attended park in the middle of a city, crows recognize individual people. Mm-hmm. That's still a lot of faces that they have to see.
Yeah, there's a lot of evidence that crows and ravens, these corvid species, are quite able to recognize individual people, hold grudges as well, and we just don't, distant birds like those that live in the Antarctic, we don't have a lot of experience around them even to know what their cognitive abilities are. I mean, we've done a lot of studies into corvid species. We haven't really done a lot of studies on Antarctic skuas, onto how, they're recognizing of any individuals. So this is something that is this is a new area. And then beyond that, the ecology of this system is that they're going to be, there's probably very limited resources. It is probably very resource intensive to be able to raise a nest of young. And if anyone is going to be coming by that nest, they're going to remember who, why, what, when. They're going to remember all the information about it so that they can protect their young but however they can.
Yeah, I think that ultimately them being able to discern that information is something that isn't necessary to defend their nest. They could just attack anybody that comes to it, but it does definitely, going back to resources and energy again, just like we were talking about in the previous story, it saves energy if they know who to attack and who is kind of cool and you don't have to worry about. So in a place like the Antarctic where it's hard to come by resources and it's hard to get energy, it would make sense that they would have to figure out how to assess threats very effectively.
And I wonder what they're using to identify the people, especially if they're, it's really cold, so people are wearing these bulky jackets and hats and maybe even a scarf that, makes the face difficult to see. Is it the way that the people are walking? Because gait is very personalized. So it could be something about the way people walk.
Or a lot of people are going to wear brightly colored jackets in the Arctic because you don't want to go missing. If you do, you want to be able to get spotted by a helicopter. So it's maybe not unlike fancy bird plumage.
Well, it's part of the study was them changing clothes, and it didn't have any effect. Okay. Yeah, but it's definitely there's something going on. I was wondering about that too because you don't have very much of your face showing when you're in the Antarctic, so. Yeah, there's something going on that these birds are able to identify humans that way, and it just goes to show you that even these animals that have never seen humans before, they will learn how to identify a threat quickly.
Yeah, and I don't know much more about the interpersonal relations of skuas and how they interact with other skuas, how they interact, because other skuas are probably coming around and trying to steal eggs or even chicks because they aren't meat-eating birds.
Right. Yeah.
So maybe recognizing individuals is helpful because they're often protecting their young from the same individuals that they see around them all the time anyway.
Right. Yeah, it had me thinking because how they identify an individual bird might be very different from how they identify an individual human. Or it might be the same. I think that's what's really fascinating to me is that when I suddenly had to learn 20 different squirrel monkey faces, I had a really tough time finding individual things to pick out. But when you meet a new person, you don't find yourself going, oh, this person has this color hair and a mole over on that cheek and blue eyes and they kind of have a frowny face. Okay, that's Steve, right?
Steve has a frowny face.
So it's just a very different cognitive process that I go through, and I would assume other humans go through when you're trying to identify individuals of a different species. So I then was putting my brain in the brain of a bird, wondering if it's similar or different.
I do wonder that too.
Yeah.
I don't know.
I don't know if we'll ever know.
Well, we're not going to answer it in this half of this show, but we'll answer more questions in the second half of This Week in Science. So everyone out there, why don't you stay tuned because we have more science coming for you in the second half. I got fecal transplants for you and your dogs. We'll be back in just a moment. Stay tuned. Hey everybody, head on over to twist.org to check out our Zazzle store. There's a link at twist.org for Zazzle, our Zazzle store, where you can find hats and mugs and t-shirts and all sorts of things so that you can proclaim your Twissy love all over the planet. Wherever you are, you can order these Zazzle products to be able to wear the Twist logo emblazoned all over. And I have artwork from Blair, and I'm going to see if I can get it up in the Zazzle store this next week. So maybe there'll be some new pretty pictures for Blair's Animal Corner in the Zazzle store soon as well. Zazzle store that's on twist.org, and we have all sorts of cool things like phone covers and hats and aprons, tote bags. Just go search. You might find something that you really, really, really didn't know you needed. But then you look at it and you go, wow, I need that Twist on me. Twist.org. Twist is supported by listeners like you. This show is currently 100% supporter, listener supported. That's right, you guys. We haven't taken ads in a really, really, really long time. And if I were to take an ad, it would only be because I knew that it would be something you would want. So I appreciate more than anything not having to deal with advertisers and being able to do this show and being able to pay our bills and being able to do what we do because you help us do it. You support us. You are the executive producers of this show, really. I mean, you're the ones who are giving us $1 here, $20 there, whatever it is. You are helping us make this show possible and to help us bring it to the world. It's all about you guys. Any amount you give, $2, $5, $500, it all helps and it all keeps this show going and it allows us to do more and more and more. We accept donations a couple of ways. We have PayPal buttons all over our website. So if you go to twist.org, look for the PayPal buttons. If you're not into PayPal, we also have a Patreon account. Patreon is Patreon. It's patreon.com/thisweekinscience. It's a crowdfunding website where you can sign up and donate on an ongoing basis, month after month, episode after episode, and we send you little gifts in return for your donation. Whatever your preference, head over to twist.org and listen to the most recent episode. You can comment on the show and make a donation. Help us out however you can. And if you can't afford a donation, you can also help us out in little ways all the time, helping us by getting more people listening to Twist and to help us get more people watching on YouTube. I'd love more YouTube subscribers. We're almost to 10,000. We need like less than 800 more people, I think. If we could get 800 more people to subscribe to us on YouTube, that would be awesome. We'd hit 10,000. That would be, yes, that's like a milestone in my book. It'll help us go to Los Angeles, and I don't know if we want to go to Los Angeles and use YouTube studios, but we could if we had 10,000 subscribers on YouTube. It'd be amazing. So we try and put the show out all sorts of different ways for you. If you can help us get the word out, help share the videos we make, share the podcasts that we make, help us get it out there, tell your friends about it. That really, really helps. Wear the t-shirt that you bought on the Zazzle store out to dinner. I don't know. All this stuff helps. It really, really does. You can listen to us on iTunes, and you can post reviews on iTunes, post comments on YouTube. There are all many ways that you can be a part of what we do with Twist. If there's something you think that we need that you'd like to help on, just send me an email. We'll see what we can start talking about. We thank you for your support. We really could not do it without you.
Believe what a skeptic I am. I can't believe you believe in that sham. We disagree, but I still give a damn. The ramification of treatments from holy men.
And we're back with more This Week in Science.
Justin, tell me another story.
Well, let's talk about ants, or rather, ants talking. University of Melbourne scientists have looked into the complexities of ant communication with the discovery that ants not only pick up information with their antennae, or antennae.
Antennae.
Antennae, but they use them to convey social signals. This is a two-way communication device. And this is a study is believed to be the first time antennae have been used to be that two-way communication device rather than just something used to pick up information from the environment. This is biologists from the university's School of Bioscience analyzed the behavior and surface chemistry of hundreds of ants to examine how they interacted. The key focus was the use and function of cuticular hydrocarbons, the CHCs they're calling, a layer of waxy buildup that covers the ant's body and that of many other insects such as bees, wasps, flies, and beetles. The CHCs are a group of multi-purpose chemical compounds that not only protect animals from dehydration, but also are used as a communication toolbox. Ants use these chemicals to identify whether another is friend or foe. According to the research, when the CHCs were removed from just the antennae of the ant, her opponents were no longer able to recognize her colony identity. This told them that the CHCs on the antennae provide information about which nest they come from. Wow.
They smell like a certain nest, kind of.
Yeah.
Yeah.
Right? And ants' antennae are their chief sensory organ, but until now, we never knew that they could also be used to send out information, PhD student Queek Wang said. Over 125 years ago, famed entomologist Augusta Farrell removed the antennae of 4 species of ants entirely and put them together. Instead of fighting, as they had would have been inclined to do, they huddled unnaturally together entirely peacefully, Mr. Wang said. So, Farrell's experiment told us about antennae being used to receive chemical signals, but our research suggests that they are also a source of chemical signals. Like everyone else, we assume the antennae were just receptors, but nature can still surprise us. Mr. Wang and his co-authors also found that CHC profiles were different depending on where on the body they were. This contradicts the conventional wisdoms that the profiles in different parts of the body, ant's body, are just the same. It's just one big covering swath of the stuff. Compared to visual or acoustic signals, we know rather less about chemical signals, and one reason might be that we are analyzing a mixture of different signals all compiled together. What we'd like to know is what else they might tell us. So there's a lot more going on, in the world of animal communication than when we talk about, human ability to communicate and talk as being this super advanced thing that we have with language. Very likely none of us could tell what town another stranger that we met is from just by their smell.
But understanding how this system works, I would love something that interferes with the communication of ants as they try to hijack my kitchen.
Right, so you make it smell or seem to them, so they would sense it with their antenna, that area is another nest's turf. And then they go, oh, the tough ants have the kitchen. We're just we'll be back over here in the garden. You guys, you keep the kitchen.
That would be some pretty clever next level stuff. I find that dish soap works just fine. Really, because they're just following the chemical trail that some sort of prospecting scout ant, some recon ant that found your little bit of sugar left on the counter or whatever it is, has found it and then left a trail back to the nest, so that all the other ants are just following that trail. So if you can eliminate that trail, soap seems to work fine, back to where the ants are entering your abode, they'll stop coming in.
Right, and it's, if I could not kill ants, I'd like that.
Yeah. I feel like, Justin, you haven't had a really intense ant battle if you think that a little soap can fix the ant problem. I think that you're due for a full-scale invasion.
All I'm saying is that I track it back to the entry points where they're coming in, and I put a little line of dish soap over there, and they stop. That's it. It's that simple. They're not.
Sure it is.
They don't have a map right? They don't have a map. And they're like, okay, if we can just get around this soap and over that wall there and climb over that and then somehow find our way down that crevice, we'll be to where the good stuff is. No, they're just following a trail. They're pretty simple that way.
Yeah, right. Well, fecal transplants are pretty simple too, sometimes.
Poop pills.
Right.
Just
Well,
take some poop, put it in a gel cap, you're good to go, right?
right. Well, yeah, sometimes. So anyway, we've learned that fecal transplants are amazing for treating treating chronic gastrointestinal disorders, disorders which lead to chronic diarrhea, chronic distress that are bacterial in nature, that are of a bacterial origin. So veterinarian Kevin Conrad of Palmetto's Southeastern Guide Dogs had been seeing many dogs with a recurrent diarrhea, adult dogs, puppies, with disorders that have led to recurrent diarrhea. He found out about the human research and he said, I wonder if we can do this. Cure gastrointestinal problems in dogs that are caused by bacteria. And often the bacterial infections that cause this they're transferred from puppy to puppy because the puppies are going around, they're sniffing and licking everything, and then
they're eating each other's poop.
They're eating
pastime.
The big puppy pastime, which is not necessarily a good thing if there's a problem like diarrhea. So, Kevin Conrad created a procedure to take stool from healthy dogs, screen it for pathogens. They took a sample of it and then let it grow on agar plates for a couple of days to see what showed up, and then identified what microbial communities grew. If the stool, the poo was fine, then they liquefied it and used a rear end feeding tube to insert it in the puppies. Basically, what they found, though, was within 12 to 24 hours, puppy symptoms, good.
Oh.
Very good treatment for puppies, that their problems, their gastric problems, went away very rapidly. He's now using the method on adult and pregnant dogs, and according to Conrad from this Ars Technica article, so what we tried to do is a fecal transplant on a pre-litter mom while she's pregnant, and she's dropping a whole litter of puppies without diarrhea issues. So now we are not only treating it, but preventing it. And now what we'll find out is, have we cured diarrhea throughout the dog's lifetime? So what they're wondering is, if they treat a mother dog while she's pregnant, will it help the puppy stay healthy throughout their life? We don't know. That's what they're looking into
that
now.
would be great to just... diarrhea gone forever.
It would be, but I mean, what happens is, when you get an imbalance, is how are you going to fix that and create a balanced system, right? The gastric problems that we're talking about, they occur when bad bacteria are ingested and overgrow, right? So taking the healthy fecal sample and putting it in, transplanting it in, basically you're saying, this is what your body should have. And you're inoculating the body to have a healthy system again. So I don't know if it is saying that the dogs will be cured. They're not going to be cured forever. I mean, if they go around eating bad poo, then this might be...
Yeah, so I think that it sounds like his next step of his research is looking at seeing at the lifetime of the animal after the treatment, right? So that, I think, is really going to be the illuminating part of this. I'm not that surprised that the fecal transplant worked in the short term. The question is the long-term effects. I think that'll be fascinating to see.
Yes. And so now another fecal transplant story came out this week. This is the week for fecal transplant stories, published in MBio. It's molecular biology open. It's an open access molecular biology journal that you can get access to if you so desire without paying any fees. Researchers from the University of Pennsylvania's Perelman School of Medicine have published their findings looking into, all right, well, we screen feces for what we know are pathogenic organisms. But what about stuff that we haven't discovered yet? Or stuff that we don't know whether it actually does good or bad, but it's just not considered a pathogen at this point in time. Does that stuff all squeaks through because we're not looking for it, we're not screening for it, basically we're not looking for it. So if a person has something, or in another case a puppy or a dog has something in their stool, has something in their stool, whatever it is going to get passed on if it is not actively screened for. So what they did is they did a normal transplanting technique, getting fecal matter and analyzing the fecal matter that was transplanted into people with chronic ulcerative colitis from a single healthy donor. What they found is that there are a bunch of things that move between the donor and the recipient, which we would expect. Not just good bacteria, but also viruses. Most of the viruses were bacteriophages, so those viruses that only affect bacteria. And that's the biggest thing. And what they say specifically is we did not see any viruses that grow on animal cells that may be of concern for infecting and harming patients. But they do say that the phages that they find, while they're not a particular health risk, they could conceivably carry toxins or contribute to antibiotic resistance in recipients. So they're saying that further research needs to be done on this as we start to implement it as a common treatment.
Yeah.
Because not only are you transferring your good bacteria, you're also transferring all sorts of things that live on bacteria as well.
When you take poo from someone, you're not just taking the good bacteria. You're taking all of the bacteria and viruses that live in their intestine.
That's right. Yep. So, good things to keep in mind.
Also, don't try this at home.
Don't try this at home, folks. Not something we would recommend here. Nope. Justin, another story?
I wonder how long it is, though, until
Dr. Justin's poo pills?
something like a Dr. Justin's poo pill is actually...
They are not yet FDA approved.
Something that you could pick up at Whole Foods, down that one aisle filled with pills.
Yes, along with the homeopathic medicines. Yes.
Yeah, I'm wondering if we shouldn't just, put the next round of TWIS dollars into funding a little startup here, and because this seems like this could be the wave of the future. We've read enough about it. We're educated enough about it. We should create the Dr. Justin poo pill.
Well, and then you can have your designer poops. You can have your Angelina Jolie poops.
Pills.
Yeah.
I'm serious. This is like a huge market. I mean, we could create and I'll do quotation marks, the laboratory tested specifications for, so that everything will be scientifically applied to this particular product. But yeah, I think we should get on that.
Take a bottle of the Tom Cruise.
No. No. No.
So my next story has a university I've never heard of and can't pronounce.
Great. So a university.
But it's Guelph? Could there be a Guelph University? It's apparently in Canada. Guelph. Anyway, I'm going to call it the University of Guelph. Professors uncovered the secret to staying as young, I guess. And it turns out it's exercise. Jeff Bauer found elderly people who are elite athletes in their youth or later in life and who still compete have as much healthier muscle at the cellular level have much healthier muscles at the cellular level compared to those of non-athletes of the same age, and of course perhaps better than younger people who aren't. The study compared world-class track and field athletes in their 80s with people of the same age who are living independently. These individuals in their 80s and even 90s who actively compete in World Masters track and field championships have won world championships. These individuals are the creme de la creme of aging. The study found that their legs were 25% stronger on average and had about 14% more total muscle mass. In addition, the athletes had nearly 1/3 more motor units in their leg muscles than non-athletes. And motor units means muscle, and consisting of nerve and muscle fibers, the stuff, I guess, that's in there telling your muscles to activate. So that it's just sort of another thing that tells you you should exercise. Now, we have learned recently that even exercise doesn't prevent you from dying in all mortality across the wide spectrum, but certainly if you are less likely to fall in older age or more able to move about so that you can live independently, you'll probably have a much better quality of life. So just yet another study that says exercise is a good thing to be doing for your body.
Nice. And you pronounced it correctly. It's Guelph.
It is Guelph.
It's okay. Not just something that you call it.
Yeah. Yeah.
No, but this goes along with, you don't want to sit all day long. Mm-hmm. And maybe exercise is good for you. So yes.
So eat healthy, protect your gut bacteria, and move around.
And move around. Do your exercise.
Mm-hmm. And don't sit. Whatever you do, don't sit. Because
it's okay to sit. It's okay to sit. Occasionally.
That's not last week's show said sitting can kill you.
So that's a little sensationalist.
Everyone, get up, run, run.
Yeah. Well, I don't know that I'm going to be actively track competitive when I'm in my 80s and 90s, but I do hope to be at least able to stand up. That's where we're going for it. A study that researchers just published in, I believe, let's see where was it? Well, Nature Medicine. There it was. Researchers have for the first time gotten corticospinal neurons to grow in the broken spinal cords, injured spinal cords of partially paralyzed rats. Now, the corticospinal column, the part of the spinal cord that comes from your brain, they're the neurons in your brain from your voluntary movement centers where you go, I'm going to reach for a pencil. Motor cortex goes bing, 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 and then it says, need to send a signal down the spinal cord and out to the arm to move the muscles of the hand and the arm to go grab that pencil, right? So the brain neuron from the thalamus goes down, sends an axon all the way down the spinal cord. Some of your axons just go to like the arm and then they have to synapse onto something. Some of those axons have to go all the way down the spinal cord to the leg before they synapse onto something. The problem is when they get injured, those axons do not easily regrow. So it's the worst part, it's the hardest part of this the hardest part of spinal cord injury regeneration has been getting these particular axons to regrow and to connect to the motor neurons, the muscle neurons that actually make things move and do stuff. We've had all sorts of successes previously in this area, but not with these corticospinal neurons. And what the researchers specifically did is they used stem cell grafts to enable the in between the injury and the new area. And so the corticospinal axons actually ended up connecting very nicely with the grafted stem cells, and then which were developing into neurons. And so they grafted the stem cells in, turned to neurons, the corticospinal axons connected to them, and then those stem cells, they also went on to connect to other things. So they filled in the lesions with new tissue. And the rats were able to move much better, stretch out their front legs to grab treats that injured rats were not able to do. So we're not at a point where it's going to be used in people yet, but these patches they think are really promising, and using stem cells might really be the way to go to be able to get corticospinal neurons to do what they need to do to get people moving again. Stem cells, yay!
They're kind of a good thing. Stem cells.
Kind of, sort of.
Blair's understatement.
Yes.
Stem cells, kind of a good thing.
Understatement of the week.
Kind of a good thing. All right, let's end this show. What are the last stories we got here?
Yeah, so sea turtles, they get caught in fishing line a lot, and a new study tells us that there could be a really easy and cheap way to keep sea turtles from getting stuck in fishing nets, and that's green LED lights costing a whopping $2 a pop. These green flashing LED lights discourage turtles from getting into the nets, but do not discourage the actual target fish species. So this could be a viable option to decrease turtle bycatch in fisheries. The one thing I will bring up that was not mentioned in this press release about the green lights is these LEDs are probably plastic, and if those fall off of the nets, that's plastic pollution in the ocean. So that's not great. So I don't know if flashing green lights installed on boats maybe could solve this problem so that they're on the boat, or I don't know. But I think that that's something that we need to keep in mind when we're putting lights on fishing nets.
Well, yeah, how permanent they are, how easily they'll fall off.
Yes, exactly.
But if they'll save a few sea turtles, then we should really consider it.
Yeah, for $2 a light, that means that the estimated cost of saving one turtle was $34. But if this was done on a wider scale, the cost would go down, of course. But that doesn't include. I was trying to do some research on the cost of a turtle taken out of the ocean, which is very hard to kind of assess. But as a fishery, when you come back to the dock, there are fines for catching sea turtles. So there's a way to figure out the cost benefit here. Ultimately, it's worth it to save turtles. So this is potentially a new option, and the fact that it doesn't let any of the animals you're trying to catch out is fantastic. My very last story is actually a bit of really good news out of India, which is that micro sanctuaries are actually extremely beneficial to survival of protected species. So when you think about, for example, an antelope species like a black buck in India that lives near densely populated areas but has huge areas that they use in the wild as their home base, as their territory, giving them a teeny tiny little sanctuary in the middle of a city might seem like it's not going to do anything. But this new research shows that it actually does do amazing things for a species, even to put aside a tiny bit of space. So any space we can give a species we want to protect is beneficial. And I think this is fantastic news.
Oh, it's wonderful.
Yeah.
Yes, and I don't know. This kind of goes along with these stories, but some recent research shows that a lot of the research that's being done into conserving species is not really being done in areas with lots of biodiversity, where the threats of extinction are the highest. So a lot of the actual conservation research is not happening in the places where it really needs to be researched.
Yeah, unfortunately, there's lots of political stuff going on when it comes to that. There's a lot of really biologically rich areas that are difficult to get researchers into. It's also kind of difficult to get the government to support those organizations sometimes, depending on the space. And then also, if you're a country that's struggling just to feed and protect its populace, it's kind of hard to tell them to fund research projects on species.
Yeah.
But I think that directly with that, I would also say that any research is helpful. We'd rather be doing the research in spaces where the animals are found naturally, but ultimately anything we can learn about these species and what they need is still going to help us in the long run.
Absolutely.
Yep.
Agreed. Research at the Korea Advanced Institutes of Science and Technology they've created a fridge laser. A laser for refrigerators.
Fridge goes pew pew!
Yeah, pew pew. And as the writer for Ars Technica said, pew pew, nom nom. It's perfect. It's absolutely perfect. So anyway, the idea is that this laser can shoot, the way a laser does, illuminate food in your refrigerator and detect the bounce back of the light. And if there's any shift in the light that would occur as the result of movement of microbes, microbes have all their little flagellae, and they're like blue, 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 flagellaing around on top of your food. If they are there on the surface of your food, moving like little microbes do, then they would slightly shift the laser light, and that would be detectable. They tested it by swabbing chicken with E. coli and B. cereus, and they determined that they could figure out which chicken samples were contaminated or infected with the bacteria and which were not. And they could determine how contaminated they were. So was it just a little bit of bacteria or a lot of bacteria? So could now we create these little lasers that scan your refrigerator once a day to determine the sanitary status of your food, and to somehow give you, or if it's a commercial refrigerator or a food processing location, could it give warnings if some contamination and how much is detected?
Yeah, so Schnago in the chat room is asking a good question. So it sounds like you'd have to leave your food uncovered?
I think, yeah, there are a few issues with this technology. That would be one of them.
Right. So I would imagine the ideal way to get this done would be to create a laser that's like a heat gun, it's a laser that you point at something and you pull the trigger and it tells you what temperature it is on the surface of wherever the laser is pointing. So something like that would be fantastic, because then you could take out your week-old dinner leftovers and crack open the Tupperware container and go, pew! You go, not today.
Right. Yeah. So maybe that's a better way.
Yeah, I would absolutely love this. I think this would decrease food waste. I think it would decrease illness, of course.
Poisoning.
Yeah. But I think food waste is a big one, is that we say, okay, I bought that a week ago, I got to throw it out. But if we can actually just shoot it with a laser and say, oh no, actually those leftovers are still good, that's going to decrease food waste. It's going to decrease money spent on food.
Yeah, absolutely. Justin, we can't hear you at all.
You've muted yourself.
Where'd you go, Justin?
It's almost as if you knew you'd say something inflammatory.
I didn't do anything. It was not.
No, Justin did it to himself.
No, you're not. I can't hear you.
Well, while he figures that out, can you tell me about chicken pox?
Yes, while he figures out his wonderfully witty comment how to make himself heard, he has to be heard for the end of the show. Chicken pox possibilities. So a lot of us get chicken pox as children, and you kind of go, yay, I got chicken pox. I won't get chicken pox ever again. Awesome. And that you think, oh, that's the benefit of chicken pox is to not get chicken pox again. Well, turns out there may be other benefits, like not getting brain tumors. So researchers at Baylor College of Medicine have found, and they looked at information from the Glioma International Case Control Study with data on 4,533 cases and 4,171 controls across 5 countries. What they found was a 21% reduced risk of developing glioma when people had chickenpox.
21% is
Yes.
pretty substantial.
Yeah, and they said that the protective effect was actually more in higher grade gliomas. So you had a less of a chance of getting the worst gliomas with chickenpox. And so there have been previous studies that have suggested this link, but this is the largest study to date that has come up with this connection. And so researchers are saying, this is more of an indication that there's something happening with chickenpox. And it's interesting because chickenpox is also related to shingles. And shingles, we know, is a disease that comes as a result of nervous stimulation. So the disease is harbored within the nerves, and then when stressful situations occur, like illness or heat or just many situations that can cause shingles to express themselves, what happens is that is virus being expressed through the nerves themselves. So nerves, brain, maybe there is what the connection is, we don't know, but this is a suggestion that there might be a role for studying chickenpox and the chickenpox vaccine to brain cancer research.
Yeah, so whoever gave you chickenpox when you were a child, you can thank them now.
Right. Thank you for reducing my brain cancer risk.
Yes. Thank you, older brother, for giving me chickenpox.
That's good. We lost Justin and he has not come back. But you know what?
The show must go on.
The show must go on. And it is the end of our hour. We did it. We have completed another show. And it is time for me to thank our Patreon sponsors. I would like to thank Kevin Parachan, Keith Corsell, Steve DeBell, Patrick O'Keefe, Jason Schneiderman, Rudy Garcia, Gerald Sorrells, Greg Guthman, Dave Neighbor, Jason Dozier, Matthew Litwin, Eric Knapp, Jason Roberts, Patrick Cohn, Chris Clark, Richard Onemus, John Ratnaswamy, Byron Lee, E.O., Bob Calder, Jared Lysette, Ulysses Adkins, Brian Condren, Jake Jones, Trainer 84, Edvardus Rimkus, Brian Hedrick, Cassie Lester, Sarah Chavez, Layla, Shane and Tara Ginsburg, Marshall Clark, Charlene Davidson Henry, Don Kamarechka, Larry Garcia, Randy Mazuka, Ed Dyer, Tony Steele, Dave Friedel, Craig Landon, Daryl Lambert, David Wiley, Robert Aston, Mitch Neves, Flying Out, John Crocker, Richard Porter, Christopher Dreyer, Andrew Dollinger, Sylvan Westby, Artyom, Pixelfly, Shu Wada, Stephen B., Dave Wilkinson, Steve Mashinsky, Rodney Lewis, Braxton Howard, Phil Nadeau, Rick Ramus, Salgood Sam, Matt Sutter, Emma Grenier, Philip Shane, James Dobson, Kurt Larson, Stefan Insom, Michael George, Russell Jensen, Mountain Sloth, Jim DePoe, Tara Payne, Alex Wilson, John Maloney, Jason Olds, James Noah Wiles, Paul West, Alec Doty, Illuma Lama, Joe Wheeler, Douglas Campbell, Craig Porter, Adam Mishkan, Aaron Luthen, Marjorie, Paul Stanton, David Simmerly, Ben Rothig, Columbo Ahmed, and Gary Swinsberg. Thank you, all of you, everyone, for supporting us on Patreon. I really want to say thank you to our new Patreon supporters who have joined us recently. It's wonderful to be able to read your name during our shout outs at the end of the show if you are in the shout out list. And additionally, to those of you who donate to us, who support us at the $1 level, and also for absolutely no reward. I know you didn't want a reward, but this is me just saying thank you, because you really help us do what we do here. So it's very important. If you're interested in supporting us on Patreon, you can find information at patreon.com/thisweekinscience. Also, remember that you can help us out simply by telling your friends about TWIS. Tell 2 people about TWIS this week. On next week's show, once again, we're going to be broadcasting live online at 8 p.m. Pacific time on twis.org/live, and you can join our chat room as you're watching the show, which is so much fun. But don't worry if you can't make it. You can find our past episodes at twis.org/YouTube and just twis.org.
Thanks everyone for listening. We hope you've enjoyed the show. TWIS is also available as a podcast. Just search for This Week in Science in iTunes, or if you have a mobile device, you can look for TWIS for Droid in the Android Marketplace, or for This Week in Science in the Apple Marketplace, including the Apple TV.
For more information on anything you've heard here today, show notes will be available on our website, www.twis.org, where you can also make comments and start conversations with the hosts and other listeners.
Do you want me to do that again?
Yeah.
For more information on anything you've heard here today, show notes will be available on our website. That's at www.twis.org, where you can also make comments and start conversations with the hosts and other listeners. Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twisminion@gmail.com, or Blair Baz at twis.org. Just put TWIS, TWIS, into the subject line, or your email will be spam filtered into oblivion. Pew pew! You can also ping us on the Twitter, where we are at TWIScience, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that came to you in the middle of the night, please let us know. We'll be back here next week, and we hope you'll join us again for more great science news. And if you've learned anything from the show, remember,
It's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got
a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness, I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
Boop, beep, doop, beep, boop, beep, doop, beep, boop, beep, doop, beep, doop, dee, doo. This is now the post-show show. Faux show. You know, no? For show. So I have sent a Hangout link to those people from Patreon who might be able to join us.
Ooh,
You
fun.
will, if they are watching now, and if you have received this message from Patreon, you will be joining a live hangout on air.
Hooray.
Hooray, huzzah. It's not a private hangout, it's public. You'll also be able to talk back at the people in the chat room. It's great. Okay, you have a pretty picture of a nautilus there.
Yes. Do you know what this is, Kiki?
Did I say it's a nautilus with pizza
It is a
on its
nautilus,
head?
but this piece, not with pizza on its head. This piece of art is the very first Blair's Animal Corner art in 2016 that is available to new Patreon supporters or old that jump back on the bandwagon, because I noticed that currently there is no one in the Blair draws you a picture item of Patreon.
That's
So
right.
if anyone who goes back in, first come, first serve, this one will be available. So the background is oil painting, and then the front, the nautilus, is actually made out of construction paper that I cut and pasted together freehand.
So can you guys really not hear me?
You
We
sound like
can-
a cyborg.
Yeah, we can hear you now. You're just cyborgian. Yeah. Okay, I need to open a window in here. It is springtime. It was like 70 degrees today, and this room is hot. I don't know what I'm going to do in this summer again. I'm going to die of sweat again. Okay, now we cannot hear you. You're no hearing. Wait, say that again? Nope. Can't hear you.
Look, here's piece number 2. Does not yet have an animal in it.
That's a nice
We'll
oil
see who
painting,
lives
though.
on this plane soon. I haven't decided yet. The canvas has not spoken to me yet.
It's so weird. Did you mute yourself? How come it went? Did you try changing the settings in the settings? Yeah. Yeah, Cylon, by your command. Right? AC and fans. It's what it's going to have to be. It's going to be the very loud room. I need to install a ceiling fan in here is what I need to do. I wonder if I can do that. I only rent. Okay, no hearings, Justin. We are no hearing. And you went Cylon, which is the weird digital problem that happens occasionally, and I think it's through Google. Am I not hearing you after you muted yourself and you're just not coming out of mute, or is it through your mixer? I don't know. Your mixer? Right, Kevin Unique is saying unplug it and plug it back in. So I have sent a Patreon message the people with a link. I haven't heard back from anybody. I don't know if anyone's going to be able to join us tonight. Ooh, I just got invited to do a segment for Dear Veronica. Yay, Veronica. I'm excited about that one. So, okay, Justin's muted now. He's going to unmute
Well,
himself.
it mutes every time.
We're back, and
Yes?
it sounds good.
Oh my goodness, this is ridiculous. Okay, so
It sounds
as
great
I
now.
was saying, I actually think this laser is going to create more food waste because the germaphobes, such as our lovely co-host, who is now a toad,
She's not a totophobe.
is going to see anything show up on that laser and toss it out. Whereas if you didn't know, you'd have eaten it and you'd have just been fine. So I think people will overreact to anything living in or on their food and throw away much more food much quicker than they do now. That was
Yeah.
That was my point.
I wonder.
That's interesting. Although I think that anything with a flagella on it is not something you want to be eating.
I don't think that's true. I think that's something that's
Yeah.
fine. People eat all the time flagellas. I have a little bottle of flagella that I like with peanut butter. But I mean I would put this in the category of you had the same laser and it was on your toothbrush and it indicated something. I bet you'd throw that toothbrush away quicker, right, Blair?
Well,
Yes.
yeah, or I would
Yes.
sterilize it, right?
Right.
I
But if
think
it's
that-
food, you're not going to sterilize it. You're probably going to throw it out.
Yeah.
Yeah, that's my point.
But I also think in America, we throw things away way too fast.
I don't know if that's necessarily true. I mean,
Maybe you don't.
well,
But I would
okay,
say
look,
the majority of people do. Especially if you think about sell-by dates and things like that, and best used by, and all those sorts of
the dating
things.
system is absolutely obscure. Best if used by, sell by, used by, best before, is it still good after?
Mhm.
I've been told by my grandmother that milk is good 7 days after the expiration date as long as it's been refrigerated. But she's also from a generation. She had 2 full-size freezers in the back because she was expecting another Great Depression. My household, I had this discussion on the Science Island hangout the other day. My household, I throw away a ton of the food. I think about a third of the food I buy for the kids actually makes it to them. Right? Because they're a little bit picky eaters. All of a sudden, they don't want blueberries, and then they want blueberries again. They have to have blueberries. So sometimes there's blueberries that go through and we're out. We need to rush to the store to get more. Sometimes they sit there and they don't get eaten. Also, with the 2-year-old, there's a tremendous amount of food that just ends up on the floor, right? So there's a lot of food waste in the household already. I don't think that knowing that the leftover spaghetti's got a little action to it that it didn't last time we busted it out, where we would have eaten it and just been fine. I think it'll just add to that.
Well, the idea is that, with science, we could find the threshold at which you get sick and identify that.
But I think that's going to be different for everybody because of their.
For certain things, but for other things like salmonella or E. coli, it's going to be bad no matter what.
Right, and so I think there's probably a better usage of this in the context of this at the supermarket, for the supermarket to utilize something like this as a check on food that maybe shouldn't go out in the first place. But I don't know. Leaving it up to the general public I think we'd just be throwing away more food. I think that would just be it. Could it keep 1 or 2 E. coli breakouts from taking place? Maybe. Maybe instead of maybe it's something you would add to that salad bar at Chipotle to help them prevent future outbreaks. But I don't know.
Let's see. So E. coli have several flagella per cell.
Yep.
So I think that's part of it too, is that they were targeting specific bacteria that are really detrimental. And I don't think that necessarily this laser is just going to find any bacteria. It's going to be specific bacteria that...
It's just going to find things that move.
It is,
Bacteria
but they were
that
using
have-
the flagella, right? So it's...
Right. So amoebas who move slowly, maybe it will not be picked up as readily.
Right.
Those that move by the extending of a foot, right?
Right. And then there are some bacteria that have cilia that move more smoothly. But the flagella just kind of causes this behavior that...
I bet cilia would cause a movement that could be detected by...
Yes, but I think
Changing
it's a different type of movement. And what I was reading in the article earlier was that it was specifically focusing on bacteria that move via flagella.
Right. But now the new and improved laser that you can buy for your home refrigerator will track 99% of all things that can grow on your food. This is the natural progression would be very quick for it to be more sensitive, tell you anything that's alive on your food. But maybe then again, this is where we're headed.
But if you eat food that has no bacteria on it, but then take your poo pills, you're all set.
Then everything's fine.
Yeah.
Mm-hmm.
Yeah.
There you go.
Mm-hmm.
I don't know.
Right, and then we can eat completely dead processed food and still have a gut bacteria that can process it thanks to the pills.
Yeah.
And then one day in the
just
future,
3D print all our food in-house.
right, just make it out of cardboard remnants.
Uh-huh.
That'll work.
Yep.
This is more and more. This is supporting my my conspiracy theory about aliens being microbiologists from the future. They're just now they work for Dr. Justin's poo pills of the future, and they're going back in their spaceship time machines to probe for better samples to recolonize the poo factory.
Ew. That's what the probes were for.
Mm-hmm. That's what it was all for.
Ew.
Yucky.
Grody. Oh, I know what I was going to open up. Dun, da, da, da. Let's see if I can get this open, and we will share results of the TWIS survey. That's what I need to share. Both of you guys took a look at that. Did you not? Yeah.
Looked at it.
I'm going to click on that, and then I'm going to go clickety-clickety-clack, and then the summary, and go, yeah, look at that. Boom! Okay, you guys, and they're in the chat room. Let's look at our survey, the TWIS survey. Is it going to help us do anything good with TWIS? So, asking whether or not people actually subscribe to TWIS, the vast majority of responders said yes, which is good. And I think looking at the reasons with it that were reasoned within why or why not, people like being reminded, and subscriptions help just kind of go, oh yeah, I get it on a regular basis, and it makes it really easy. And then a lot of people, most people still get it via podcast. And the duration of the podcast, so talking about show duration, the duration of the podcast people prefer is 30 to 60 minutes is the largest percentage, but we also have a very high percentage of over 1 hour. So,
yay, long form.
Well, but don't you
Long
think that's
form,
kind of
so-
self-selecting? Is the people taking the
people
survey are the ones
are-
that like our show as long as it is?
So
I believe you have a point there.
yes. However, Blair, if
Yeah.
what we're talking about in expanding our audience is finding more people like those who already found it, then
Sure.
this would be the sample that would be.
But we
most
can
pertinent.
also look at the fact that more than half, right, of our listeners. Now I want to look at it again. More.
Oh, I'm sorry. I'm not even putting. I have to put it up here so you can see it. There we go.
Yeah. So almost half of our listeners want it to be 30 to 60 minutes, which means if we're going to get more people, there's probably more of those people than anybody else.
Oops. Right. Then there's more. Right. There are more of those people than others. Yes. Mm-hmm. So something within the 30 to 60 minute range is good. In thinking about YouTube, though, it's interesting. The majority are kind of in this 5 to 10 minute to like 15 to 30 minute.
Pretty even spread, yeah. The biggest number is over an hour.
Right. Yeah, actually, you're right. The biggest number there is over an hour. Mm-hmm. But these are, again, maybe the people who are watching TWIS on YouTube, which is pretty awesome.
Right. But so and then part of it, so there's a difference between there's a difference in these audiences of whether you're looking for going in to find 5 to 10 minute bits of stuff on YouTube, maybe because you've only got time for a couple quick looks, or that you're just scanning around watching a bunch of different shorts on things. But the over 1 hour category is telling you that for these people, YouTube has replaced that sort of sit down in front of the television time, right?
Right.
They're
so
no longer
using it like that, yeah.
Right. So there's people who are utilizing it as people have utilized television in the past, where you turn it on, you sit down, and it's there in the room, whether you're directly focused on it or going about doing your other stuff. It's just on playing a program in the foreground, background, whatever. It's there anyway. The 5 minute people, I think, typically are looking for a certain type of info or infotainment, and then scanning through some stuff, or maybe just jumping into YouTube for a bit, looking around, and then moving on. I find, and this is why I've always sort of talked about preferring myself the long term or long form on a YouTube, is that I do use it like a television. I put it on, and now it's on. I'll watch a YouTube program, or it'll be in the background while I'm cooking and doing something else. So I do mostly podcast type stuff, but good informational stuff too. And so for me, it would be annoying if the content changed every 5 minutes, and partly because a lot of times you would end up stuck in commercials. If you're going around trying to watch different things, at the beginning of each one there'll be this commercial, and it can be 5 to 10 minutes can work if it's on like a play all sort of a section, which I don't think we have yet. But if you hit a play all, even if somebody has broken up their podcast into 5 minute segments, you go through the commercial, maybe the skip ad, maybe the first one, but the play all, the rest of it can show up commercial free. And so then it's fine if you're going into a segment or a YouTube channel that's a segment of it could be music, for instance. I was looking up different surf guitar bands and stuff and then people have these playlists, and you click on one, and each one's a couple minutes, but it continues to play through them. So I don't know. Depends on how people are utilizing it.
Yeah.
I think it's a good idea to have shorter segments, but I think it's also a good idea to have them running together sort of like a theme. So within This Week in Science, somebody could click into Blair's Animal Corner, and a whole mess of Blair's Animal Corners could play one right after the other.
Right. Like, so if we were to get them edited out and create playlists of Blair's Animal Corner. Yeah.
Or the short science news at the end of the show is probably also something that people would want to hear. The quick stories, right?
Yeah. So something I'm just saying to get to that question that what do people listen to? The majority say they watch or listen to entire episodes, and if only part, then it's the first half. But then I asked about the second half. If it was only Blair's Animal Corner, only interviews, or other, and then other was giant, and I don't know what other is.
That other's probably the after show.
Right? Yeah. Yeah, so the first half of the show is the part that people will start watching, and then if they drop off, then they drop off from watching it. So,
But that's if only a part of the show. So I mean, I think that one sort of says right there that the only reason somebody leaves the show once they started watching it is because they had to. It's not that they're skipping ahead to or
right,
just
because
looking.
the majority are like just watching. 70.6% said they watch or listen to entire episodes.
Yeah.
So yeah, which is awesome.
And then
that makes me so happy.
Right, but then the other huge chunk, 24%, was also sometimes yes, and sometimes no. Meaning they all intend. Pretty much everybody intends to watch the entire show.
Sometimes you can't.
But then it doesn't always finish.
Yeah. Cool. Most answers about what people wished we did more of revolved around physics and space.
And interviews. People like the
interviews.
They want more interviews.
But a few people I noticed too also said that they liked interviews as standalone things as well.
Right. So if we put those out as their own individual things.
Yeah, that'd be a fantastic segment for that YouTube channel, is just the interviews, right?
Yeah, no, there are all sorts of good recommendations coming out of this. Rambling. This quotey voice. Most people love it as it is. Just kidding. Face mites. Some people don't want any more face mites. I can't say that. There were a couple about religion and making it more discussion.
One wanted me to stop bashing religions in general. Another one said we should do more of it. And
yeah.
Yeah, and there are like this reporting results that nobody on the show understands. Every once in a while we think that we've got a story that we're like, yeah, we got this. This is really interesting, and this is going to be an interesting discussion topic. And then you go to talk about it, and it's just you don't explain it right, or I don't know. We all just sit there and go, huh. The excitement and the discussion and the understanding. Sometimes
I think
we don't do it right.
That's the gamble, though, of having a live show, and that ultimately, like, we could rehearse everything beforehand, and everything could be canned, and we could know what we were going to say, but then the payoffs when we do have an insightful conversation would not be as high. And I feel like ultimately we get a lot of amazing conversations, not to toot our own horns here, but like I like hearing stuff from both of you guys about things that I didn't think about and then exploring that topic. And I think that means that every once in a while, maybe 9 times out of 10 we have a good conversation, but 1 time out of 10 nobody has anything to add. Whoops.
Yeah. And I can be responsible for a decent amount of that too, especially in the last quarter, I would say, of the show. I start to become a less active listener. Start to get very distractible.
Well, that's good because the show's been going on for a while and we've been talking about stuff.
I'm also fidgety, and then I kick.
Are you saying the show shouldn't be as long, Justin?
I'm saying the show's way too long.
That's good. Yeah, it's good. But we got a lot of really great responses, and it's pretty awesome. I think from here, like, I don't know whether I think we'll stick with this survey for a while. I don't know. We might use this to ask some other later questions.
Right.
I don't know. Figure out more specific stuff we should do with it. I don't know. Some great responses in there.
So
very
helpful. I
Yeah.
think what's important to note in all of this too is that we can do all of it because we have a pretty long format. we have a long enough format. I mean, we have unlimited time available, but we use a good hour to two a week putting the show together. For for the long form listeners who like this, also the sort of behind the scenes, the after show conversation stuff, we have all of that content. It's all there. Perfect. Boom. We put that out there. And because we have already the long format, it's easier to take out segments, shorter segments of the show, and spin them out there, so that we can have 5-minute segments and 10-minute segments or 15-minute segments of the show broken down. That's doable. Well, the tough thing would be if we had a 5-minute show and people were asking for an hour-long show. We'd be like, how do we come up with a whole hour? That would be
Right.
really
How
hard.
do we do that?
Yeah, we'd have to do the show we'd have to be doing it 10 times. And it's too difficult, right? We're already in long formatty live show. That's the hard part, right? Everything else is an easier version of this. And everything else, the content is already created. It's annoying that we have to edit. That's the whole horrible, tedious, painful part of it. But it's a lot easier to do that than go from a 5-minute show to, or think of the contrary. Like if we're doing our hour and a half, and somebody was like, "Really, you need to be doing 12 hours." Right? That's what the difference would be. Right? Really, you need to be doing a 12-hour show. That's what people really want. That would be really hard, but we're already at the long form. If we were doing this an hour a day, right? If we did this show an hour a day, I think that would be the next that's sort of getting it into the longer format where we have a long format and a lot more content of it. That would be the next thing.
That would be rough.
But as it is, I think we've got segments. I like the part somebody said in there we need to do more of the shtick, more like Blair's Animal Corner, more of like the Kiki's Casper CRISPR Cas9 sort of segmented stories. I keep imagining I'm going to start doing more of This Week in Science History. I think I did one not too many weeks ago, but
I feel like you always have This Week in Hominids.
Yeah, I've been sort
Human
of on.
history.
I've been working on the human history angle for sort of
This Week in Your Family Tree.
right. Yeah, the origin story stuff's always fascinating. There's always I'll pick up anything Toxoplasma gondii related. But there is
old stuff.
There is a good
World Robot Domination.
There is a good point that This Week in the End of the World. There is a good point that we don't, and we never really have to a great enough a degree focused on space. Space works its way in, but we don't. I think we have the best interviews on space. I think we focus on those quite a bit. I think we do really good finding somebody else to talk about space.
Yes.
That's what I try to do.
Yeah, because
we don't bring a whole lot of it.
Space.
Yeah. And this is why, years ago, I tried to get a guy who was studying physics to be a co-host on the show. He was great for a little while, but it didn't work out long term. Because I knew that my background was not the hard sciences, physics, and astronomy. So I was hoping that someone else could kind of bring those stories. But, I love the mix that we bring right now. And I do try to bring the physics and the space stuff, but unfortunately I don't know it as well.
We touch on it.
The experts do.
So this is also, I think, okay. I think if you had somebody, Michio Kaku is one example. Who is a physicist who understands these things very, very well. And as a communicator, when I hear him talk about these subjects, I walk away realizing I've learned nothing. Because he's developed a way of talking about physics that can be completely digestible. and as such, you can't learn anything from it. And there's an element of this, I think, that's a problem in science communication, and I think we can do a decent enough job of breaking down that there's something really interesting going on without absolutely being able to digest it and understand it ourselves, and still bring it without being afraid of not being thoroughly able to vet the subject matter ourselves. And especially if we're talking about quantum physics, which if you talk to two different people who are quantum physicists, they might both say that they actually don't really get it themselves, but they're working through it too.
Yeah.
Right? Like, there's some really intensely conceptual versions of reality going on there that have actual reality-based things that the science is discovering and working on, but it is not going to be something that you can talk about without just talking in complex numbers. It's not, there's no easy analogies out there.
Yeah.
But we'll try.
Yeah. So for the time being, until I'm able to, I don't know, get more editing done. The time being, I'm going to stick with putting the time codes in our YouTube descriptions so that at least people who go to YouTube will be able to skip through to different stories, individual stories and segments, if that's what they so desire.
Yeah.
And that'll be a pretty easy, clickable thing to do for people. And then the next step after that is to hopefully be able to get to a point where we can start editing things and scheduling more. I'll try and schedule more interviews, maybe like at least one a month. Try and do something like that where it's somebody from physics or space sciences to discuss something so that we get that into the show.
Yeah, I think that would be awesome.
And maybe have them not just be an interview like we've done before, but also like we've done with other people like Physics Girl, have them join the show and bring a few stories.
Yeah, that's always a blast.
Be like a co-host for the hour.
Yeah.
We also interview them too.
I love doing that. I also like it even if they haven't brought a story, but they hang out and comment along with the stories. That's always fun.
So that's like an easy place to start, and then we can hopefully start expanding to getting more things edited, and do that before we expand into doing other shows.
Did you know that Twitch TV has your JTV videos back on their service just like JTV had them?
What? No. What? I don't even...
Twitch.tv/Dr. Kiki.
Did Twitch
They must have
buy
absorbed.
Justin?
They must have absorbed them.
Because I did not ever open a Twitch TV account.
But if you had a Justin TV, I guess there it is.
That's interesting. Really?
Interesting.
I'm gonna go look for that right now.
I know. Loading.
Loading. What's loading?
But I definitely suggest, Kiki, looking at some podcasts on YouTube. There's a few that I follow.
You guys, I have a whole channel.
There's a few that I follow that have done. They'll do the long form. Will show up as a podcast, and then at some point later, they'll have that same podcast broken down into smaller segments, like sort of the subject matter segments within that podcast.
In a podcast, they have it actually where you can skip to sections?
No, they don't. They don't have time codes. They don't have skipped segments. What they do is they put out the full thing, which can be like, an hour and a half, 2 hours, whatever it is. That comes out first. Then later, it's that same episode, but broken down in like 5 to 10 minute segments or a half-hour segment, depending on how long the subject went. So they sort of then just cut out and post those segments afterwards.
I wonder if people would hate that, like getting bombarded with like the same show over and over and over.
Well, I don't think you're getting bombarded by it. It's sort of like you can then just go to instead of it's sort of like what you're doing with the time stamping, but instead it's already separated out. And so it also creates if somebody did sit and do a play all, and the play all button, I don't think we had a play all button on ours, but if you hit the play all, you can go through all the short segments, and then at the end of those short segments, it would go to the full episode. So you could either skip all those, go right to the full episode, or start like, I don't know how much time I'll have, I'll just do short segments, and then you can just leave it off there whenever you like. And if it goes all the way down, you've gotten to the full show. You can either listen to it again, or you can just decide I'm done.
Right.
Interesting.
But then you're also, instead of getting one viewer for the full show, on the segments, you're getting a view for each one, which looks like a larger amount of viewers overall. But it also means that people can not have to watch the whole show, but we sort of get credit for what they did watch of it.
Right. Something I was just thinking about too is that we don't do a lot of it, but we do definitely reference earlier moments in the show and other segments throughout the show.
Mm-hmm.
And sometimes topics return, and
right.
I definitely think it makes more sense when you listen to it all at once.
It does. I totally agree.
Certain topics, you gotta listen to them. I think I should get a mohawk. I live in Portland now. I need colored hair and a mohawk.
Mm-hmm.
Don't you think? Hey, Justin, I think you need to go pirate scuba diving.
I'll find Davy Jones' locker and I'll bring it back to the surface, I said, says I. See.
I wonder how that will go. Where'd that sound effect come from? That was awesome. Sound effects are good. How do I get this? There we go. A monkey.
I could put glasses over my glasses.
Crickets.
Seems unnecessary.
I would like to be in Amsterdam right now. No, this is Venice.
That's
I'd like to
Venice.
be in Venice right now. Here we go. Look at my amazing background. It's blurry.
So I don't know if anyone actually got invited to this hangout or not.
I tried. I sent a link in. I sent a message and sent a link to the Patreon people who should have gotten something, and says I sent it to 10 people. Well, I want to see what 10 people it was. I know. We're sitting here. I try to do this. I try. Where are you? Are you in the ocean, Justin?
At the fjords.
The fjords.
Fjords.
Fjords. No, I went back there on purpose. Loop-de-loop. One. Yeah, maybe the people who would have gotten it are asleep right now because
Possible.
many of them are on the East Coast. So tomorrow I'm going to have, not tomorrow, Friday I'm going to have the hangout. Justin's floating. He's like a disembodied
decapitated
I'm a
head.
disembodied decapitated.
You're decapitated. I need a new face. There we go. Meow, I had to go to the beach. Meow, right meow.
Right, meow.
Ah, that be the fjords a talking again.
So I'm gonna try another Patreon hangout on Friday at noon Pacific time for those individuals who are able to come to that one. And I was talking with Chris Clark, who is a donor. We were talking about other times
Chris
to have
Clark,
it.
Chris Clark, who is the well, you can't see it now because it's hiding, but he's the person who sent me this boom mic.
That's right. He did send the boom mic. Yes.
Justin, you're killing me. I'm trying to hear Kiki.
And what he would like, he's on East Coast time, and there are other people I'm sure on East Coast time, but who work. And so for them, 4 or 5 p.m.-ish is probably optimal, but on like a Friday. But could maybe, I don't know, it'll be hard for me to do much later, but like 6 or 7 p.m. on a Friday, is that doable for Blair?
All right, I just muted Justin. Oh no, he unmuted himself. Gonna kill you.
What's that, Blair?
Nothing. Okay.
Okay.
Yeah, Friday works.
Friday at noon Pacific time. Friday at noon Pacific time.
What? I did not understand that. I thought you said 6 or 7.
No.
I'm a little distracted. I wonder why. I'm having trouble following.
And so the hangout this week is Friday at noon Pacific time. The hangout that we're talking about having could potentially be a Friday at like 4, 5, 6 p.m., 7 p.m. Pacific time. Could you do like a 6 p.m.?
Yes.
Yes?
Yes.
What if any earlier than that, or is 6 p.m. good?
It depends on the Friday. Are you talking about this Friday? No.
No, a
Okay.
different one.
So, yeah, depending on the Friday, I could get off work early. I could go to work early and get off early, and I could do earlier than that, for sure. I could do potentially as early as 3 or 4 o'clock if I was given enough notice.
Cool. All right.
But noon, I might be able to jump in the chat room on my lunch break this week.
Oh, cool.
So
Nice.
maybe if there's a chat room. Will there be a chat room? Will it be on air?
It will not be on air.
It will not be on air.
Pirate Justin. It will not be on air.
Okay.
No.
Because I don't.
It'll be a private hangout, a private Patreon hangout.
Mhm.
I think it should be live.
I won't have
think
a camera at work, but I would be able to watch and then respond in a chat room. I could do that.
the pirate says it should be live, or at least recorded so we can post it to their YouTubes.
Sometimes I don't want things posted to the YouTubes. It's a
I
very
know, but
casual thing, you know.
It will be, but it's okay. This is a casual show. We can call it a
just a
casual
casual-
Friday show.
It's a casual hangout. I don't know.
We can even label it the casual minion hangout. Like, it's okay to have casual stuff.
It is okay to have casual
This is
stuff.
the new media.
and
The new media is made up of casual people doing professional things. Professionally
Turn
casual.
this off.
Hey, are you trying to... Can I...
Remove all effects. There we go.
Oh, where'd the cat go?
Where'd the kitty cat go? I don't know. I got distracted. Oh, now he's got sunglasses. What?
Ah!
Um,
Actually, that mustache looks pretty good.
Identity 4, the hangout is not usually live streamed. I usually just do it as a hangout, and it's just a private Patreon hangout. If people are okay with live streaming, I'll make it a live streamy thing. But I thought that people just wanted it to be a hangouty kind of thing, you know? No. Oh, no. Anyhow, yes. What donor level get hangout invites? Those at $10 and above get the hangout invite. What else? And what else? Who, what, when? Going back to the comments about the Twitch stuff. Got the comments about the Twitch stuff. It's interesting that they have merged it all over. Justin TV started the Twitch, and then I wonder if they are trying to get old users back. And at one point in time, I had lots and lots of views because Justin TV was promoting it, and so when I was doing it, like all these people came in and viewed and took part in discussions. It was crazy for a little while there. And then Justin TV was like, no, we got to do the gaming. We're only going to do the gaming.
So, did you record a voicemail message for this person that pledged $25?
No, because I asked Justin to.
Oh.
And Justin has not.
Oh, yeah, I need to do this.
Yes.
Or just give them $25. I have to choose.
So Identity 4. I do not believe that you are borked, but you are
Borked.
in. He's wondering if his Patreon is borked. Bork. It is not borked, but you are in the no reward donation area. So, if you're at the level, I don't know. Yes, you're at the level, so you definitely could. And I will invite you. I'm sorry that I did not invite you tonight, but you were in the no reward section. Sometimes people do this, and I don't know if they're doing it on purpose or if they have done it accidentally.
Ah.
Because sometimes the Patreon system, it gets a little confusing as to what you're actually signing up for. So...
Send me the details of this people I'm making a message for. I need to figure out how to do a message that doesn't sound too ridiculous, that they wouldn't use it.
Yeah, I think he wanted like a disclaimer.
Yeah, okay, I
kind
can
of
do
message.
that.
Yeah.
I just need the name of who I'm doing this for.
Mm-hmm. Okay.
I'm going to guess Kiki sent that to you before.
You know, it's too bad I turned off. Where's my sound effects? Hang on.
Oh, where's my mute button again?
Yeah.
I need to have that one handy. Hi, Blair.
I'm muted again. Oh, Identity 4 can't go to the noon one. Duh. And you're standing here right now, and we're thinking of going home and going to bed. You see that head nod right there? I saw that Blair head nod. Because we all went, how come there's nobody here?
Honk shoe.
We've been here for 45 minutes and nobody wants to hang out with us. Except the people in the chat room. You guys are hanging out with us. You're awesome. But, so I'm going to have to make up for Identity 4. Hopefully. We'll figure it out. Maybe we'll have to do another one next week. If I can. Maybe another one next Friday. Fridays are a good day for me.
Stand by.
Afternoon hangouts.
April 8th.
I got nothing going on April 8th. So.
I'm at a climate change workshop at the Exploratorium.
That sounds like fun. Identity 4. TWIS night. You're busy recording TWIS. Me too. Blair's Reindeer Corner. What?
Someone was saying
to Finland to do Reindeer Corner.
So Janisky wants us to have Science Island in Finland, and
That
someone
would
said
be
it was too
awesome.
cold and there was too much snow. But I said, but then I can have a pet reindeer.
It's awesome. Yes. Ben. Yes. You're like, I want to hang out. Yeah, I know. I know. Gotta figure
Oh, our chat room has
something out.
worms, you guys.
What? What do you mean our chat room has worms? Oh yeah, it does. At the very people in the list. Worms is in there. Hi.
Worms. We got
Our
worms.
chat room now has worms. Awesome.
Mm.
Oh, when is my stem cell podcast? So I recorded the first episode of the stem cell podcast today, and it should be released on April 12th, I believe, is the date that they have planned for release.
What is this?
I have been hired, I guess. I am a co-host now for a podcast called the Stem Cell Podcast.
Cool.
And it's stem cell focused. But
they're kind of important, right?
Yes, stem cells. They're kind of important.
That's what I said before, right? Stem cells. They're kind of important.
What happened to Justin's video?
It's still here.
Kiki, I think you should have me on the show to say obvious things.
To say obvious things. You'll just be in the
Hey,
background. Hey guys.
Stem cells. Aren't they kind of important?
Kind of important? What? Yeah. Oh, there's worms. Worms are saying hi. You know.
Yeah. Uh-oh.
Hi-ya. Hi-ya. That's what pinworms say in the middle of the night. Hi-ya. Hi-ya. Anti-worms threats. No worms. What? You promote growth. That's fantastic. That's fantastic. Gunshots. Identity. Hasn't this happened before? Didn't this happen another day? Didn't we tell you to drop and cover? Drop and cover, Identity. No gunshots. Oh yeah, Hot
Wait.
Rod, you're right. We need a user named Bird now. That would be awesome.
Identity, don't you live in Washington State? I didn't know they were known for their crime rate up there.
It's hardcore up there. Gotta tell ya.
Apparently.
I know. I'm reading backwards through the chat list. Orcs, you're cracking me up. With Wisconsin guy gonging Jackson Fly. Nice. That's what we need to do. More gongs. Exciting neighborhood. That drop and cover. If you hear gunshots, drop and cover.
Really.
Low. Get low. Let me hear low. Aha! Yay! You fixed your Patreon. Okay.
Oh, good.
Uh-huh.
Now I want to go look.
Mm-hmm.
Oh my. I'm getting tired.
Yeah, you're getting tired.
I thought you were in Seattle. I just didn't want to. I wasn't sure if you wanted to tell everyone what city you lived in. I didn't anymore.
He's been outed as a Seattleite.
I was trying to be sneaky.
Mm-hmm. So let's see. Let's look at the calendar, because we have to look at the calendar every time we do a show so we can determine whether there are things that we're looking forward to. April Fool's Day on Friday. You should maybe not trust anything that I post on Friday.
Or me.
Except maybe the link to the show. And then we have Square Root Day on Monday, April 4th. So I'm not going to go to the dentist that day for a root canal. That's not going to happen. Then we have Yuri's Night coming up April 12th. That's fun. And then we have Earth Day coming up. And so the 20th of April, we're supposed to go green and be earthy. So we'll have a themed up show for that. Did we have something? Were we going to do get an interview? No? Yes?
For which?
Earth Day?
I don't know.
I know I wanted to get an interview for World Day for Laboratory Animals.
Mm-hmm.
So it wouldn't actually be Laboratory Animal Day that we would do that interview. It would more likely be on World Tapir Day. that we would do that.
Love me some tapers.
Gotta love the tapirs. Yes, it's a big April. It's a big month. Some good days in there.
I think Identity 4 might have just bought my Nautilus. We'll see. You'll have to let me know.
Oh.
Uh-oh.
Mm-mm.
If anyone else wants that Nautilus, you'll have to get in the $15 reward now and duke it out.
That's right. You gotta get in there. Get in there.
I have more coming, though.
Haha. Going green was March 17th, says Ben Ruthig. I wore green that day. And I posted my video about the green dye in the Chicago River. Yeah, Identity 4, I think you might have been in no reward for a long time now. I'm glad you fixed that. Fix it! Do we have world... Not tapeworm. I said tapir. Taper? Tapir?
Taper?
Not
It depends.
tapeworm. I don't want a tapeworm day.
Depends on where you are. In America, we say taper.
Taper. Okay.
But in, like, Brazil, they would say tapir.
Zoo. Yes.
What's that?
Quantum Club.
Oh, right.
Quantum Club. Oh, Twit Refugee. Shh. Portland. It's awesome. Portland rocks.
Haha.
Shh, but don't tell anybody I said so. Where did Justin go?
I don't know. I'm still here.
Come back so we can say goodbye. I can't say goodbye while you're not even here.
Goodbye, everybody.
There you are. Oh my goodness.
It speaks.
All right.
Rude.
Oh, you guys.
So
All
very
right.
rude.
It's so rude, so rude.
Yes.
Yeah. I'm excited. More shows next week. It's gonna be good. I'm gonna edit this here podcast up tomorrow. I'm gonna get this podcast edited, and then we are gonna be good to go.
Yeah.
Yeah.
I'm falling asleep.
I know.
Me too.
We're all
I'm
very
super
tired.
faded.
Faded.
I will see you two next week.
Okay. I like
Good night,
that.
minions. Good night, Kiki. Good night, Blair.
Good night,
minions.
everybody. Have a wonderful night. Have a wonderful week. We will see you next Wednesday. I do hope you'll be back in the chat room again or back watching us live. Have a great one. Bye.